All right. I would also like to thank the organizers for giving me this great opportunity to highlight some of our work at this celebratory symposium. Um, um, Dr. Goodnow and Dr. Nemezi's work has really been inspiring for me and continues to inspire the work that we're doing in our lab currently. So I'm going to talk more about my research is more fundamental in nature as compared to the two previous speakers of today. And I'm going to talk about B cells of early life origin. And I hope at the end of this seminar that you'll be convinced that the early life B cells are actually very interesting to study from the perspective of understanding B cell tolerance. So yes, so the adult B cell compartment, both in mice and in human, is a mosaic composed of long-lived B cells that are self-sustaining and that may be of early life origin, and more short-lived B cells that are continuously replaced by new B cell output from the bone marrow. And one very clear example of this is the presence of B1 B cells in the adult mouse B cell repertoire. So B1 B cells are innate-like B cells in function. They are the major producers of natural IgM in a mouse serum, as we heard from Frida in the previous talk. And they preferentially develop from fetal precursors and are not efficiently generated in the adult, which preferentially generate conventional follicular B2 B cells. However, they are self-sustaining and perform a non-redundant function in the adult immune system. And these observations inspired the layered immune system hypothesis put forward by the Herzenbergs decades ago now. And this work was primarily done using transfer experiments such as this one from Hardy and Hayakawa, where they took fetal liver precursors or adult bone marrow B cell precursors, and transfer the cells into an immunocompromised recipient. And as you can see, the CD5 positive B1 B cell compartment was efficiently reconstituted by fetal progenitors, but not from adult bone marrow. However, these experiments, although this pointer is not doing what I want it to do, these experiments were instrumental for our understanding of the developmental changes in B cell output, but doesn't tell us so much about sort of how much fetal B cells actually contribute to the adult B cell pool in situ during unperturbed development, and also doesn't tell us so much about their actual developmental window. So in order to address these questions, we wanted to perform lineage tracing of early life origin B cells in situ and observe map their fates in the adult mouse. So to do this, we used the MB1 CRE-RT2 CRE driver, first developed from Mikkel Reth's lab. And MB1 is encoding for this is expressed in pro-B cells early during B cell development, and expression persists later in mature B cells as well as plasma cells. So by inducing CRE expression using tamoxifen, we can label the B cells that have developed or are developing or pre-existing at the time of induction, and then follow these B cells across longitudinally across developmental time. The B cells that develop later on after we administer the tamoxifen will not be labeled, so that we will be able to distinguish their developmental origins. So to first show that the model works. So 3 days post tamoxifen induction, we could see that a majority of follicular B cells get labeled, become TD tomato positive. However, 12 weeks later, you can see that the follicular B cell compartment has largely turned over and been replaced by tomato negative B cells. On the other hand, the B1 B cell compartment, which is we know is of early life origin, is not replaced and remains time-stamped. So we could then use this model to separate early life origin B cells from those developing in the adult mouse. So with this model established, we've spent the past 8 or so years in my lab to really learn as much as we can about these early life origin B cells. When do they develop? Where do they go? What do they do in the adult immune system? And also how their development is regulated during early life. And today I will touch upon a couple of these topics. So in order to understand sort of the actual contribution of fetal on early life B cell development in the adult immune system, and also understand their developmental window, we went ahead and systematically time-stamped the mice at different time points, pre and postnatally, early in life. And then importantly, we waited 3 to 4 months before analyzing the adult mouse to understand sort of the developmental window of the early life B cells that we can find that constitute a portion of the adult B cell pool. And when we looked in the CD5 positive B1 B cell compartment, which is the prototypical early life origin B cell subset in the mouse, we could see that certainly there are 20% of the B1 cell compartment that is time-stamped already prenatally. So the adult B1 cell compartment, around 20% of those B1 cells actually come developed prior to birth. However, we also saw that more than half of the B1 B cells actually emerged after birth within the first 2 to 3 weeks of life. And this is different than what was previously thought, which was that B1 B cells were primarily of fetal origin. We instead highlight the neonatal window of being quite an important time for B1 cell output, and at least for the cells that stay on and end up persisting into the adult. What was very surprising to us initially was also that we found that IgA plasma cells in the small intestinal lamina propria, a large proportion of those are also of early life origin or come from B cells of early life origin, and they actually emerged during quite a strict window between day 5 and day 10 postnatally. And this window actually, thinking back about it now, maybe is not so surprising because, of course, the postnatal period also marks the exposure to, external environment and the microbiome, and may correlate to the B cell activation that happens following the disappearance of maternal antibodies and the intake of solid food during weaning. So we had a look at the lamina propria, and this was done, by the way, in collaboration with our session chair, William Gase's lab. And you can see that the time-stamped tomato-positive plasma cells nicely line the lamina propria of the small intestinal villi. And these are overlapping with the IgA staining. And these plasma cells are also actively maintained by B cells of early life origin in the germinal center of the Peyer's patches, the chronic germinal centers of the Peyer's patches, which is the, of course, inductive site of IgA in the gut. So in essence, a lot of the all of the antigen experienced IgA B cell subsets that we looked at were enriched for early life origin B cells, and this their representation in the adult was sensitive to antibiotics treatment. So when we treated the mice with 4 weeks of antibiotics, you could see these representation of the early life B cells decreases, in line with the idea that these cells are reacting to and maintained by the microbiome. And what about sort of like more specific ways of looking at early life memory in this fraction of B cells that are time-stamped? So to look at this more carefully, we took advantage of a rotavirus infection established by Katarina Lau's group in Lund University, who we collaborate with, and we infected the mice at day 5, time-stamped them to mark the early life origin B cells, and then 9 to 12 weeks later, we looked at the rotavirus-specific IgA plasma cells, and we could see that a majority of these are residing within the time-stamped fraction. Just proof of concept that time-stamped B cells do contain neonatal memory, antigen-specific memory. What about in the IgM compartment? Well, we also tried to immunize the mice with group A streptococcal heat-killed group A streptococcus, following a protocol developed by John Kearney's group at University of Alabama. And similarly, so this immunization is known to induce the expansion of a glucnac-specific B cells actually residing within the B1 cell compartment. And we found the same result, and we could also see that also these glucnac-specific B1 cells also are largely time-stamped, residing in the early life fraction. So in essence, we think that the early life B cell pool is an archive for memory B cells generated early in life. Oh, sorry, the circles plot looks a little bit funky here, but imagine a circle around this. But what's also interesting is that neonatal immunization can generate clones that cannot be found if you immunize with the same antigen in adult life. So both in the case of rotavirus, we found the VH13 encoding clone that was recurrent when you immunize neonates, but not when you immunize in the adult. And similarly, Kearney's lab has demonstrated before that group A streptococcus reactive, glucnac reactive clones is of a different clonal composition when the mouse is immunized as neonates as compared to adult derived glucnac-specific clones. So we think that the neonatal period is a window of opportunity to actually recruit unique clones into the long-lived memory B cell pool. And so I have shown you that the adult B cell compartment is actually consists of antigen experienced B cells that are primed potentially early in life and persist into the adult to contribute to adult immune responses. And we're currently hard at work to understand the role of these early life B cells in adult antibody responses and in the regulation of adult immune homeostasis. But what I also wanted to show you a little bit more today is the work that we've done to understand how these B cells are selected and the unique developmental program that is restricted to early life that actually shapes the neonatal B cell repertoire in a unique way. So I have worked since my postdoc time on a RNA binding protein called Lyn28B. It is a microRNA binding protein that binds the stem loop region of the precursor LED7 family of microRNAs. By binding to the LED7 precursors, it inhibits the LED7 biogenesis and blocks basically the LED7 microRNAs from being generated, such that the expression of Lyn28B actually leads to the derepression or the overexpression of LED7 target genes, which include c-MYC, PDK1, and ARID3A are some important examples of target genes of LED7, which are basically their expression is increased by Lyn28B expression. And we and others have previously shown that Lyn28B is necessary for the normal development of B1 cells early in life. And when it gets turned off in the adult, B1 cells, our development is attenuated. However, when we overexpress Lyn28 again in the adult setting, we can reinitiate the output of B1 cells. And what's interesting about B1 cells is, of course, that they are self-reactive, and they're known to be positively selected by a self-antigen into the repertoire. So in the adult situation, B1 cells, which are self-reactive, are excluded from the mature B cell pool. But this seems to be breached somehow. This checkpoint seems to be breached somehow early in life, and also when Lyn28 is expressed later on. So Lyn28 expression, so we made a reporter mouse, which nicely showcases the kinetics of its downregulation after birth. So Lyn28B is highly expressed during fetal life, and as you can see in the hematopoietic stem cells and the B cell precursors, as well as mature B cells, the expression is rapidly downregulated after birth, such that it basically is completely turned off by day 19 post-birth. And this window, of course, as you might remember, overlaps with the output of early life B cells that I showed earlier in the lineage tracing study. And so to start to probe how Lyn28B might affect sort of the positive selection of self-reactive B cells, we started looking at CD5 expression. So CD5 expression is like a crude marker, you can say, for self-reactive in mouse B and T cells. It has been used previously to study positive selection during thymocyte development. However, it's not expressed during B cell development in the adult. But interestingly, we found that CD5 is actually expressed during neonatal B cell development in the immature B cell stage, right after the assembly of a mature BCR. And in particular, we see that CD5 expression is high in this phosphatidylcholine reactive immature B cell subset, which is a known self-reactive specificity that is destined for the B1 cell lineage. So having observed this, we then started using CD5 as a readout to start to interrogate changes in B cell selection early in life. We could see then that actually CD5 expression is, as I showed before, quite highly expressed during B cell development in immature B cells day 2 after birth, but gets slowly downregulated and gets completely turned off by day 19 to become indistinguishable from adult immature B cells, suggesting that this window really correlates with what we saw for Lyn28B expression when it became turned off. And indeed, the CD5 expression pattern is controlled by Lyn28B. As we can see here in day 2 old mice, their immature B cells, their CD5 expression is completely dose dependent on Lyn28B. So in heterozygotes, CD5 expression is lower, and in knockout mice, they are even lower, resembling what we find in the adult immature B cells. And this also correlates with a dose-dependent decrease in the output of CD5 positive B1 cells in the peritoneal cavity. And the opposite is true when we overexpress Lyn28B in the adult setting. When we do this, when we turn on Lyn28B for using a doxycycline-inducible model for a week, we can see that CD5 expression is induced in the immature B cell stage. And here we also used co-staining of CD5 in a NER77, a reporter model. So NER77 is a reporter of antigen receptor signaling in T and B cells. And you can see that it really positively correlates with CD5 expression. And NER77 is not really expressed in the pre-B cell stage, but is induced in the immature B cell stage significantly by the expression of Lyn28B. So this really strongly suggested to us that Lyn28B actually promotes the positive selection of potentially more self-reactive specificities early in life. And I should also acknowledge the work from Richard Cornell's lab at Oxford, who actually used the MD4 HEL model, a BCR transgenic model, pioneered by Professor Goodenow, and showed that in the presence of Lyn28B expression, shown in brown, that antigen, the presence of a self-antigen allows for the accumulation of B1 cells bearing the transgenic receptor. And this is different from in the absence of Lyn28B and mimics the situation that you get from neonatal derived B cells. So altogether, this really suggests to us that Lyn28B does license the positive selection of self-reactive specificities. And so we know from Dr. Nemazi's work that the major central tolerance mechanism in B cells is actually receptor editing. So might Lyn28B be controlling receptor editing and thereby allowing for changes in the self-reactivity of B cell output? And here's some ongoing work in our lab, but we do find some evidence that this might be happening. So when we overexpress Lyn28B in adult mice, we do get a decreased percentage of lambda light chain expressing immature B cells, which is consistent with a decreased receptor editing, since the lambda light chain is one that's sampled after attempts to recombine the kappa light chain. And the opposite is true in the neonate, where we have, in wild-type neonatal situation, we have low lambda usage. But in the absence of Lyn28B expression, we get a dose-dependent increase in lambda usage, consistent with increased receptor editing and more stringent tolerance induction mechanism. Altogether suggesting that Lyn28B might be limiting the extent of receptor editing and thereby allowing for the output of potentially self-reactive B cells early in life. And we know a little bit about how Lyn28 works to do this. So we know that Lyn28 upregulates MYC target genes and actually looked at MYC expression, which is also increased 2 to 3-fold. And we know that LET7 is a negative regulator of MYC, so this is expected. And we also found that Lyn28 increases protein synthesis in pre-B and immature B cells. And in fact, this is of course in line with the upregulation of MYC, since MYC increases ribosome biogenesis and protein synthesis in the cells. But we also found that Lyn28B can directly actually interact with mRNAs encoding for ribosomal subunits, and perhaps thereby also directly promoting ribosome biogenesis. And so we think that this molecular pathway may play an important role in the promoting of the positive selection. So to summarize everything that I've shown you today, this is our working model. We think that Lyn28B is a unique molecular program that controls and shapes the neonatal B cell pool such that it has an increased cell-free And this unique, sort of functionally distinct B cell pool is then imprinted by external antigens as well as self-antigens to induce long-lived early life origin B cells that are maintained in the adult immune system. And we're, as I mentioned before, very interested to understand their function and contribution to maintaining tolerance as well as immunity in the adult immune system. And with that, I would just like to thank all the people that have contributed to this work. So Stefano Vergani was a former postdoc and did all the lineage tracing work. Stein, as well as Hugo, are also former lab members that have been instrumental in understanding the role in the selection early in life, in B cell selection, as well as Elena, who also is currently working on our little receptor editing project, and our collaborators, of course, and funding. So happy to take your questions. Thanks.